Hello and welcome back to Microbiology. Today we're going to talk about human microbe interactions. Really what this chapter is going to be about is infection, which is whenever microbes get in, they attach, they reproduce, and then they cause disease. This can result in a pathologic state, which is a cumulative effects of infection damage. And this really leads to disruption of tissues and organs, resulting again in disease. But really, disease is any deviation from health. Naturally, there are lots of things that could cause disease, including infection, which we'll talk about, diet, genetics, even aging. But as we said before, our main focus is infection here. So we'll do infectious disease, which is the disruption of tissues or organs caused by microorganisms or their products. Before we get too far into disease, we should point out that we do need bacteria in and on us. In fact, we call the good bacteria occupying space in and on us the normal biota. There's a lot of different things that would naturally live in and on our body. We often call these things resident or indigenous, but we can have things like transient organisms that are moving in and out. This will also include not just bacteria, but fungi, protozoa, and even viruses. So one of the bigger science projects that's been going on in more recent history has been the Human Microbiome Project. And really what this is a study to figure out what organisms people have and where. Having a good biota, we understand at this point, makes you healthier. So understanding what healthy people have versus what non-healthy people have could really give us lots of information on how to protect people. Early results have shown that there are significant amounts of microbes that live in and on us. Roughly, again, we think the products made from these microbes even help us in our functions as humans. And so ultimately, really, we're learning a lot about what we have and where. There are even some places that we thought were once sterile that may actually have microbes living in them. The other important thing that we've discovered is that really the biota's presence has helped with overall health. And so the makeup of your intestinal biota can greatly influence really all aspects of health. So having good gut bacteria allows for good, healthy, basically digestion. But again, having poor gut microbes ultimately leads to potentially things like Crohn's disease or obesity, heart disease, asthma, autism, diabetes, moods. The idea here is that the microbes have a bigger role in our health than we appreciate. And so this is actually good news because we could actually encourage good bacteria to live in specific areas and promote, again, greater health. So currently we understand that there are lots of places where microbes would live. So definitely places that we know we have lots of microbes include things like the skin, the upper respiratory tract, the gastrointestinal tract, the typically outer portion of the urethra, external genitalia, vagina, external ear canals, and again, the eyes. Basically things exposed to the outside. Some other things where we're thinking we have a lot more biota include things like the lungs. For a long time we thought the deepest parts of the lungs didn't have a lot of bacteria routinely living in there, but we're starting to think that there's some. Again, the bladder, breast milk, amniotic fluid in the fetus. Again, we thought traditionally these things were very sterile. But really, where we think we should see very limited amounts of microbes making up a biota would be in places like the brain and bloodstream. There are, again, many microbes that will travel through the bloodstream, but they're considered to be more transient. So as I've been kind of stressing before, that having good biota actually helps with overall health. They actually encourage appropriate development of organs. Again, they help really prevent the overgrowth of bad things. And it's because of the process called bacterial antagonism, where again, good bacteria compete against bad bacteria, not letting the bad bacteria grow out of control. We did a C. diff example in the last chapter that really illustrated kind of what we're talking about. That as long as we have good bacteria in our colon, C. diff doesn't grow out of control. But naturally, one of the things that will always be antagonistic with even our biota is the immune system. So one of the bigger things tied to whether infection is brought on is really level of immunity. Unfortunately for us, there are several things that affect immunity, including things like your age. And whenever you're very young, you're building up your immunity. Whenever you're very old, your immunity is breaking down. We also see things like genetic defects, where again, genetically, you are more predisposed to have a weaker immune system, or even acquired defects like AIDS, for example. Pregnancy is another situation where naturally immunity goes down. Surgery or physical trauma also decreases immunity. Additional disease like cancers or other infections could lead to, again, greater risk of infection. Using drugs that hurt the immune system, like immunosuppressive drugs, or even literal stress can cause issues with immunity. Endogenous infections are going to be things that are already present causing infection. The idea here is that normally your immune system would keep them in check, or the environment they're in wouldn't favor the chance of infection. So whenever the environment changes or the immune system goes down, now things that are endogenous or already living there could cause infection. A good example would be like E. coli getting into the bladder. Again, E. coli usually found in the colon and not an issue, but whenever E. coli gets in the bladder, it grows quite well and out of control. The resulting situation would be an infection called a UTI. One of the more important things we're starting to understand is that our initial colonization happens really at birth. And so it's important to understand that the more effectively we initially colonize, the greater chance the offspring has a healthy life. And so one of the things I want to point out is through natural birthing processes, the first microbes the baby would be exposed to are the microbes of the mother's vagina. As a result, again, we would expect certain microbes to establish early on in development. But again, if we do something else like a cesarean, we can have a situation where we're not being exposed to the right microbes that would help set up a good, healthy initial start. But we've learned that really we also continually build or alter our normal biota, and so things like breastfeeding has been well documented as containing lots of resources like specific bacteria and even sugars that babies can't break down. These are for the microbes, by the way, to encourage, again, healthy microbes to develop in the gut. So the idea that, again, vaginal birth and breastfeeding would lead to a natural, more healthy biota. I do want to stress that both my children were born via cesarean, and both my children were partially breast and partially bottle fed, and both of them are actually incredibly healthy. So we do understand that this is going to be kind of a start process, but do understand that we see the biota is something that's continually changing over time. So I'm in no way saying that you can't have a healthy baby if it's born via cesarean or even bottle fed. But what we are starting to see is science supports that the child has a greater chance of being healthier if they, again, are born vaginally and are breastfed. So I really don't need you guys to learn where every bacteria is going to be found and all that. Don't worry about that. But there are some things that are kind of expected. Like we find staph on the skin. This isn't the first time we've talked about this, right? Whenever we even talked about mannitol salt agar, we pointed out that staph grew on the skin where there's lots of salt. Or like E. coli grows in the colon. It's called E. coli for the colon. Okay, so the idea here is there are some things that are expected. But if you don't know what Bacteroides is, you sure as hell shouldn't really stress about where do we find it. So as I said before, again, don't stress on these things. There are some things that are given, like strep, you would expect to be found similarly commonly in the oral cavity or, again, throat. But most of the stuff you don't have to stress about. So let's go through kind of the progress of infection. But as we go through, we're going to use some important words. So we should probably define them. The first one we're going to define here is pathogen. And that's a microbe whose relationship with the host is parasitic. And so the result would then be infection and disease. So literally, the growth of the microorganisms hurt the host. That's what pathogens do. Whenever they grow out of control, they cause infection that hurt hosts. There are some things that are far more likely to cause infection and disease, and that is true pathogens. True pathogens can even cause disease in very healthy people with normal immune systems. Opportunistic, on the other hand, is something we've been kind of describing before, which is this idea that if a microbe's moved to a different place or the human's immune system goes down, now we have normal microbes, like the endogenous, that can now cause disease. So a new word to point out here is opportunistic. So opportunistic illustrates that if microbes are moved or the host immunity goes down, now normal biota can cause infection potentially. So one of the things I want to talk to you about is the fact that not all microbes are created equal. In fact, we put them into categories based off the risk of working with them called biosafety levels. This is put forth by the CDC, or Centers for Disease Control. And the idea here is that whenever we go through these levels, which are 1 through 4, as the number gets higher, the risk in handling and working with the microbes becomes higher as well. So if we look at this, notice biosafety level 1 is kind of our lowest. Again, very low chance of getting infected by working with these, and even then, not going to cause serious illness if you do contract it. Okay, some examples include things like Micrococcus or Bacillus megaterium or Lactobacillus, even Saccharomyces. Saccharomyces being things like baker's yeast. But notice whenever we go up to level 2, things start to get a little more serious. We start to see pathogens like Staphylococcus aureus and E. coli, Salmonella, diphtheria, hepatitis A and B, rabies is level 2. And so the idea here is we're ramping up the risk quite a bit. As a result, we should have at least some safety precautions like lab coats and gloves and glasses, and have the ability to work in cabinets if needed. And that's the minimum things to be properly capable of handling these things. And so the idea, the reason we harp on you so much in the laboratory about wearing your safety gear, is we work with potentially BSL-2 pathogens. And so the idea is we need to take those steps to protect you. So please wear your safety equipment in the laboratory. At level 3, we start to see a bigger change in, again, potential risk. Now we're seeing things like, TB. TB is level 3. Tularemia, caused by tularensis. Yersinia pestis is literally the plague. Coccidioides immitis, again, another pretty potentially bad pathogen. But one of them that typically shocks most people is HIV. HIV is just level 3. And here we also, and whenever we work with these, notice again, we're going to do most of our manipulations in safety cabinets. Again, we're going to make sure that we have special clothing. We're basically going to make sure that everybody's vaccinated and protected against infectious dangers. Again, we're ramping up the safety because the risk has gone up. But really, level 4 is the stuff of nightmares. With level 4, we see lots of things that will straight up kill you. And whenever we look at them, they'll do it quite quickly and easily. These are all going to be viruses that have, again, high morbidity and high mortality rates. So whenever we look at these things, now we're not just playing with just everything in cat. We literally have buildings designed to work with these organisms, where you literally have to change clothes and shower before you go in and after you come out. Literally nothing can leave or enter the room without making sure that it is safe to do so. Literally, fumigation and autoclaving of all the equipment is absolutely necessary. The next word I want to introduce to you is virulence. This is really our degree of pathogenicity. The more virulent something is, the more likely it is to start causing infection and cause serious damage. Naturally, anything that increases the risk of causing infection or causing damage, we call a virulence factor. One of the things you have to understand is you kind of have to take in a certain dose or inoculum before you're typically likely to get sick. And so we like to put a number on that. And so what we call that is the infectious dose, or the ID, which is the minimum number of microbes required for causing infection. As a general idea, the lower the amount of microbes required, the more virulent the thing is considered, the more likely it is to make you sick. Okay? And so the idea here is if something has a low ID, it's often pretty dangerous. So let's go through now the progression or steps of disease. So notice we're going to go through these steps starting with finding a portal of entry. the microbes have to get in. Once inside, they need to attach to allow for a surface to reproduce on. Naturally, if they're capable of reproduction and surviving host defenses, they start to cause damage. That damage we now call disease. And as a result, the body becomes an unfavorable environment for them, and so they want to get out through the portals of exit. So let's go through each one of these steps, starting with step 1, the portals of entry. This is typically the pathway through which microorganisms get in to cause infection. Many times it's through the skin or our mucous membranes that line our gastrointestinal and respiratory pathways, even our genital pathways. Basically, any hole to the outside world should have mucous membranes. Exogenous versus endogenous are two important words. Exogenous means the infection comes from the outside. Endogenous, on the other hand, means the infection was already present in or on the body. So how can we get past the skin? Well, many times it's through nicks, abrasions, punctures, or tiny holes that develop. In fact, ultimately, the skin is one of our most protective organs we have because it's a pretty tough barrier to actually allow for microbes to get through. And so naturally, if we have holes in it, then we can have pathways in for infection. A common portal beyond the skin is the gastrointestinal tract. Naturally, since we eat and drink a lot of stuff, we're taking in with the food and water things that are pathogens. But we do have a protective strategy in the gastrointestinal tract, and that's the use of digestive enzymes and really significant pH changes in the stomach. By having those barriers, we can decrease the risk of infectious pathogens causing disease. But by far the most common portal of entry is going to be the respiratory pathway. The respiratory pathway is one of those pathways that are used continuously. From the day you're born to the day you die, breathing is the number one thing you have to do. And as a result, we need ways to protect the respiratory pathway. And that comes in the form of continuous mucus that's being produced. Naturally, this thick mucus will often cover the upper respiratory tract, the sinuses, and even the auditory tubes. The idea is we're going to use the mucus as kind of like a sticky trap for the bacteria. But one important thing to understand is if we don't get mucus out, then the microbes readily grow in it. As a result, then you could have serious infection. The next thing I want to talk about is the urogenital portal of entry, which is going to again be sexual pathways. So many of these things are sexually transmitted infections or STIs. Ultimately, there are going to be huge numbers of things that are only transmitted by sexual means. And believe it or not, it's roughly 4% of all infections in the world are actually STIs, which is quite an alarming number. Again, there are roughly 13 million new cases in the United States every year. So how do we get in through the sexual and urogenital pathway? Well, through things like the penis, the genitalia on the outside, the vagina, cervix, or even the urethra. An important thing to note is that whenever the baby is being developed, we want to protect it from typically pathogens. And so we put a lot of emphasis in trying to screen and protect the baby. And so one of the important things that has to develop to protect the offspring is the placenta. The placenta is ultimately an exchange organ that allows us to filter out or control what's moving to the fetus. And that way we can make sure only resources are going to the baby and not bad things like pathogens. I do want to point out that some pathogens can and will pass the placenta, and those are the ones that we're really concerned with in terms of exposures during pregnancy. As I was just illustrating, we are concerned with certain pathogens coming through, which is really categorized with TORCH here. TORCH stands for things like toxoplasmosis, other diseases, rubella, cytomegalovirus, or herpes. The idea is it's never a good time to get AIDS, but during the development of offspring is definitely not a good time. Once the pathogen gets in, the next step is really to attach to the host. So adhesion becomes a really big important process for the microbe to cause infection. Ultimately, depending on what features the microbes have will determine how easily they can attach. but once attached, they can start reproducing and invading other areas. Another important thing that affects where a microorganism is most likely to attach is that quorum sensing, which again, quorum sensing was that chemical messaging to encourage specific microbes to grow in specific places. So here are some illustrations of some of the things that would lead to greater adhesion by pathogens. Things like fimbriae, capsules, or even again spikes on viruses. Once we've attached, we now have a nice surface to allow for growth. They're naturally going to call this surviving host defenses, but again, these go hand in hand. The idea that you can't reproduce if you're being killed by the host defenses, right? So again, I like step 3 to be reproduction. Ultimately, phagocytosis is a big concern for the microbes, so any feature that prevents phagocytosis is very helpful. So antiphagocytic factors, like your capsules, would again help the microbes cause infection. So notice step 4 is going to be the accumulation of damage leading to disease. So we call the features that allow for damage the virulence factors. So ultimately, these are adaptations that hurt the host. And so naturally, these are problems for us and cause signs and symptoms of disease. Good example is exoenzymes. Exoenzymes are going to be things secreted by really microbes like bacteria, fungi, protozoa, and even larger things like worms. The idea is that they're going to damage tissues by typically breaking down things on the tissue or parts of the tissue. A good example are things like keratinase. Guess what keratinase is capable of doing? Well, breaking down keratin, which is an important fiber that helps, again, with the structure of many tissues. Another great example would be toxins. Toxins are different from exoenzymes in that these are chemicals produced with the intent of hurting other organisms. So we have exo and endotoxins. Exotoxins are again found on the outside of the cell, while endotoxins would be found on the inside. But how could endotoxins hurt us if they're inside something like a gram-negative bacterial cell? Well, whenever that cell dies, all of its internal contents become external, including the endotoxins, which now can interact with our tissues and cells and cause issues. An important thing to understand is that the damage caused by microorganisms isn't the only damage that can cause us injury. Literally, how our body responds to the threat can also inflict injury upon us. Again, if the immune system responds too excessively, it could potentially even kill us. So let's go through where we find infections, starting off with localized infections. A localized infection will start in one point and typically remain in that place. Good examples include things like boils or even warts. Systemic infections, on the other hand, are going to spread all around throughout the body. A great example of this is chickenpox. Once you get chickenpox, it's not going to show up in just one area, it's going to show up across the body. Bifocals is an interesting topic where this will typically start off in a localized location and then move to a systemic style situation. A good example is TB. TB will start off in the lungs, but in late stages of tuberculosis, we see the infection spreading throughout the whole body and causing issue to a variety of organs. One of the other things to note is that many infections aren't just one pathogen. Most infections are actually what we call mixed, where multiple things are working together causing the infection. We would often refer to some of these things as polymicrobial diseases. A good example would be gas gangrene. Gas gangrene comes about whenever initially a form of Clostridium starts causing infection in deep tissue, but really then multiple organisms get together to start consuming you as basically a giant smorgasbord. As I loosely just illustrated there are some infections that start out with just an initial pathogen, but then quickly additional infections come on. We call the initial infection primary, and then any additional that follows up secondary infection. two things we should probably point out is the acute versus chronic infections. Acute should be basically short-lived, while chronic should persist over a long period of time. Now let's talk about really warning signs of disease, like signs and symptoms. Signs are very different from symptoms because signs are going to be things that can be measured or observed. Symptoms, on the other hand, are going to be feelings. So again, we can't measure nausea. So nausea would be a symptom, but we can definitely measure a fever. A fever would be a sign. It takes both signs and symptoms for us to form a diagnosis. We would call this diagnosis a syndrome. So here's a list of signs and symptoms. Again, things like fever, septicemia. Can we see microbes in the blood? Again, evidence of infection due to chest sounds. Those are observable things. But again, symptoms are feelings. You feel malaise or fatigue. Again, you feel itchy. The idea is, again, symptoms are feelings. Inflammation is typically one of the first signs that really show up for infection. The idea is inflammation is typically going to be characterized by things like swelling and redness and heat and pain. This is the body's attempt to try to eliminate a potential infection. This can lead to, again, things like edema, which is an accumulation of fluids. Granulomas or abscesses, where we're trying to wall off or even fight potential pathogens. Or even swollen lymph nodes, which illustrates, again, the immune response trying to kick in to fight infection. There can be signs of infection in the blood, including leukocytosis. Leukocytosis is going to be an increase in the levels of white blood cells. This would illustrate that the body's trying to fight back against a potential infection. Leukopenia would be the opposite, where we're losing a fight against an infection, and thus we see a decrease in white blood cells. Septicemia is going to be a general state in which microbes are multiplying in the blood and are present in large numbers. Again, we often refer to these patients as being septic, and as you're probably well aware, that's a pretty bad place to be. With bacteremia, on the other hand, we still see bacteria in the blood, but they're not actively reproducing, and they're typically at smaller levels. Thus, again, we're not quite as bad off as we were with the septic state. Viremia is the presence of viruses in blood, and it doesn't really matter whether they're reproducing or not, so we simply just call viruses in the blood viremia. I want to point out that some infections can go unnoticed. We often refer to these things as being asymptomatic, subclinical, or inapparent infections. The patient is infected but does not show the signs or symptoms of disease, or at least very, very minor signs or symptoms. And as a result, again, they're not aware they're infected. One of the important things to understand is once the body starts to become an unfavorable environment, the microbes tend to want to get out of the body then. So we start seeing them moving towards portals of exit. Portals of exit are going to be the pathways out, which includes basically all the opposite reactions from the pathways in. So instead of swallowing down a bacteria, you would be vomiting up a bacteria. Instead of breathing in a bacteria, you'd be coughing or sneezing the bacteria out. Secretions, discharges, slough tissues, these again are examples of portals of exit. So respiratory and salivary portals of exit include things like mucus and sputum, nasal drainage, and even, again, moist secretions. The idea is anytime we're hacking or coughing something up, we're trying to get pathogens out. Skin scaling is another source of lots and lots of potential pathogens. The idea is the outer layer of skin is, again, dead and constantly being shed. But every time you lose cells from the surface of your skin, you actually shed millions of bacteria as well. I do want to point out, most people don't realize, but the biggest component of household dust is actually skin cells. But on those skin cells, don't forget, there are huge amounts of bacteria. The fecal exit is pretty self-explanatory. Crap happens. The urogenital tract, again, this is commonly seen with the STIs where we see things like discharges or even, again, secretions. or it even can come out as part of the ejaculate. The idea is pathogens can be carried out during sexual contact. The other place that we again see large amounts of potential pathogens leaving is during things like UTI or kidney infections, whenever things are discharged through the urine. Another common source of potential infection would be again blood or bleeding, as individuals that are ill can have microbes leave them through their blood. One area of concern that is quite serious actually is that of the blood-feeding animals like ticks, fleas, and mosquitoes. The idea here is they can feed on an infected host and then pass the infection on to the next host that they feed on. And so the idea is we can have active transmission even though we weren't trying to do so. So we conveniently call those things vectors. So vectors carry things, and in this case ticks, fleas, and mosquitoes are disease vectors. One of the things I want to stress is this idea that many pathogens can actually become latent or just hang out in a dormant state. During this dormancy, again, we're typically just waiting until we get an opportunity to cause infection again. We would call this opportunity the reactivation of the infection. The idea is typically when immunity goes down, those dormant infectious agents come out to play. We also see squalia, which is again long-term damage that comes from infection. So a good example would be something like the side effects or symptoms that come from a Lyme disease infection. Since Lyme disease has an affinity for cartilage, it will often cause damage to the cartilage, which results in arthritis. But it's important to understand that cartilage really doesn't repair well or heal very well, and as a result, those signs and symptoms associated with the arthritis tend to last even beyond the infection. There are 4 phases of infection and ultimately disease. It starts off with the incubation period. This is whenever you are infected, but it takes time for the first signs and symptoms to show up. Then we enter the prodromal period, whenever we start seeing our initial signs and symptoms. Most of these things are typically pretty vague. At the period of invasion, or the height of infection, we typically see lots and lots of microbes and then lots and lots of ultimately signs and symptoms. This is usually the time whenever you're most likely to be able to be diagnosed with a specific disease. And really at this point 2 things can happen. You either die or you get better. If you get better, you go into the convalescent stage. In convalescence, the infection goes down and thus the signs and symptoms go down. So here's an illustration that's showing us exactly that. Early on, even though you're infected, you're not really showing signs and symptoms. Once we see the prodromal period, as we're starting to see exponential growth, we start to see the initial signs and symptoms. But again, these are very vague symptoms. Things like nausea or fever, or again, things like malaise, right? Again, nothing specific. But then as the infection keeps increasing, we start to see a significant increase in symptoms as well. And as we said before, there tends to be this kind of breaking point at the height of infection, and you either die here or things start to get better. And so you can see here the immune system is starting to kick in, signs and symptoms are leveling off as microbe numbers are starting to level off, and then we go into that convalescent period where things start to get better. There is a situation where potentially you get all the way better, or we see the microbes start to plane back out as they start to overcome immune response again. The next set of words are going to be important in understanding the kind of concepts of where infections come from. Starting off with reservoir. A reservoir is a primary place to find a pathogen. These things would be continual, so like water, soil, plants, or even some animals. The idea is this is places you can routinely go to find the infection. A source, on the other hand, is going to be somewhere that has basically picked up the infection and will be a short-term place to now provide the infection to someone else. And so a source is a short-term place to find infection. Reservoirs will be long-term. So here's an illustration of those. Again, you can take a look at it. but for now, we'll move on. So let's talk about carriers, because carriers are a common source of infection. They even may be potentially reservoirs if they're a long-term source. So let's point out the different categories, like asymptomatic. Again, asymptomatic, this individual has no idea they're infected and typically won't take the steps to prevent infection. An incubating carrier is someone that's still in the early stages of infection. Again, not knowing they have infection, they're not going to try to protect others by preventing the spread. Convalescent carriers are those people that are now feeling better but still can pass the infection to others. We have the chronic carriers, where again they do have the illness but don't have signs or symptoms, and so typically can pass the infection off. But I want to point out with chronic carriers, they have this kind of in a perpetual continual state. So they have the infection, but they do not show signs and symptoms and do not really get better. And so the idea is they can continually pass it. The passive carrier, on the other hand, is someone that never actually becomes infected, but instead picks up the infection and carries it to other people. So we already defined vectors a little bit ago. A vector is something that's going to carry something. But in the case of disease, we're going to talk about disease vectors. And these really come as typically arthropods or even some insects. I want to point out that there are two different types of vectors, biological vectors and the mechanical vectors. A biological vector actually participates in the life cycle of the pathogen or promotes it and allows it to reproduce inside of it. A mechanical, on the other hand, is not typically going to do this, but instead is just merely going to transport the infection to someone. So here's illustrations of biologic vectors. Again, chickens actively allow for the growth of salmonella inside of them, and as a result, they're a common source of salmonella. Another good example of a mechanical would be something like a cockroach. The cockroach will get into areas and pick up pathogens, but it's not actively trying to support the growth of the pathogen. Instead, it'll just carry it from one area to another. Zoonosis or zoonotic disease is going to be a disease that initially starts in animals, but then is transmitted to humans. Yes, I know we are animals as humans, but we're talking about lower animals to higher animals. The idea here is typically we are the dead-end host and do not continually push the microbe onto others, so zoonotic diseases really have to come from the initial source, the animal. Again, this can come from simple just contact with an animal or the consumption of the animal. So there are lots of examples of zoonotic diseases. Things like rabies, for instance, are often transmitted from animals to people. Or even things like anthrax may be carried through livestock to people. So the next thing we need to talk about is a non-living reservoir. If you recall, a reservoir is a common source for infection, and really there are just three non-living reservoirs for us: soil, water, and air. Ultimately, you can always find microbes that can cause infection in all three of these places, and as a result, we should try to protect ourselves reasonably from infection. One of the things we should point out, though, is that diseases can actually be spread from one person to another. We call these diseases communicable disease. That person that's capable of spreading the disease to someone else is called contagious. A non-communicable disease is a disease that cannot be passed from person to person. Instead, we'll have to go back to the host source or reservoir to get the infection. Whenever we talk about how disease can be transmitted, we can have horizontal transmission, which is again spread from one person to another, or vertical, which would be passed from parent to offspring. The idea is, again, horizontal is from individual to individual, vertical is from parent to offspring. Vehicles and fomites are an interesting topic. A vehicle is an inanimate material commonly used by humans that can transmit the infectious agent. I want to point out that the vehicles tend to often be reservoirs. So common vehicles include basically soil, water, and air. Fomites, on the other hand, are inanimate objects that harbor and transmit pathogens, but not are a continuous source of infection. So this would be like the tabletop or your cell phone. There's not enough resources on the tabletop to usually keep infection going. And as a result, again, it's a temporary place to pick up the infection. A common pathway that we see disease transmission is through the oral fecal route. It's exactly what it sounds like. That by some means, fecal material enters a person, causing potentially infection. As I said in the previous slide, water, soil, and air are common vehicles for infection. And pathogens can come out of humans into the soil, water, or air, and ultimately be taken back in by other humans from these vehicles. Droplet nuclei are a great example of what we're talking about. Droplet nuclei are dried microscopic residues created when microscopic pellets of mucus and saliva are ejected from the mouth and nose. This is very similar to aerosols, which is going to be a suspension of fine dust or moist particles in the air that contain live pathogens. Either way, a human is depositing pathogens into the environment that then can be taken up by other humans in the near vicinity. A commonplace infection can be passed to others is in the healthcare situation. And we call these infections healthcare-associated infections. Basically, these are diseases that are acquired or develop while in the hospital, typically with another illness. The idea is the frequencies can be quite high. Anywhere from 0.1 to 20% of all admitted patients should acquire an infection while in the hospital. One of the things to note is medical asepsis. This is a practice that should lower the load in patients, ultimately allowing for decreased risk of infection. Ways we can do this is again using that aseptic technique. Naturally, surgical asepsis is far more serious because we are going to penetrate the host's defenses, so we're going to take the extra steps to make sure that we're not going to pass infection. These techniques to prevent the spread of infection are typically developed and enforced by the infection control officer. They're in charge of literally training the workers as well as tracking potential outbreaks and any breaches in aseptic technique. Koch's postulate was an important postulate that really showed us cause of disease. And in fact, Koch was the first person to really prove disease through his postulates. So let's illustrate what his postulate was. Ultimately, he would take and introduce the potential pathogen into the host. And if it caused the disease, he would then pull it from the host and put it back under scrutiny and then back into a new host to prove that it was the cause. Epidemiology is going to be an important topic of really microbiology, because what epidemiology does looks at the effects of disease not just on individual patients, but on the large scale of community. In order to do this, we need to know how much disease is out there. So there are reportable diseases. These things we call the notifiable diseases by law must be presented to the individuals that would need the data to make determinations. And so really, by law, hospitals are required to report to local and federal authorities rates of infection. So again, typically they basically pass the information to things like the Greene County Health, and then Greene County Health can then scale it up, pass the information to the state or even the CDC. The idea is that by tracking disease nationwide, we can get a bigger idea as to what risks are out there. And these risks are actually all published by the CDC in a weekly morbidity and mortality report that tells you frequencies and locations of current infections. Ultimately, we would like to see this happening at the world level, and so the CDC actually does share the information with the World Health Organization so that we can actually understand risks associated around the globe. So let's learn some epidemiology terms, starting with prevalence of disease. Prevalence is the total number of existing cases with respect to an entire population, but you need to define the population. So like how many people have HIV in the world. Incidence, on the other hand, is the measure of new cases over a specific time period. So like how many people caught gonorrhea in Springfield last week. I want to point out there's another name for incidence, and that's morbidity rate. So morbidity is the number of new cases. This is different from the mortality rate, which is the number of deaths in the population due to a specific disease. So here are some graphical representations of data collected through epidemiology. So notice in like letter A here that we've seen an overall downward trend from 2000 towards 2010 in cases of hepatitis. But since 2010 on, we've seen a pretty steady increase, and that really is troubling to epidemiologists because it suggests that there's failures in either our methods of protecting people against hepatitis or the general public's knowledge. We don't just track disease though. Notice in B, this is the rates of influenza vaccines in different age categories. And as you can see, it stays pretty consistent. That really, overall, about half the population will get the vaccine, and thus about half the population has some protection against the disease. Down here in C, notice we're graphing rates of malaria. Basically, red is bad, yellow is still bad, and then green means there's no real cases of malaria. But notice if we didn't graph this, we wouldn't pick up this trend that right along the equator we find the highest rates of malaria. And it's because really the cause of malaria is a protozoan, but that protozoan is carried by a vector that is a mosquito. That mosquito tends to live around the equator. So naturally you can go through and look at several other examples of these different data representations, but as long as you understand we can use the data to learn more, that's what I care about. But we should talk more about the terms like point source and common source. A point source is one in which the infectious agent came from a single source. So that's why it's called a point source. There was one point that was the source of the epidemic or outbreak. A common source epidemic is going to be something that routinely happens. We should see routinely huge numbers over a given period of time. So really this image over here shows you a good example that we usually see potentially a huge spike in this particular time frame. And so this was showing specifically the American Legion Convention that led to Legionella. So we know that the exposure was a common exposure. A propagated epidemic is going to be huge numbers of infectious agents that are being passed person to person in a society over time. And so the idea is typically it started with one person, but now it's being passed person to person because it's a communicable disease. A pandemic is a different type of epidemic. This is going to be whenever an epidemic is spread across continents or across the world. The idea is an epidemic is bad, but a pandemic is really bad because now it's not just affecting one population, but multiple populations. So here's some more terms that you should probably know, including index case. An index case is the first patient found to have a very particular disease, and as a result they're the source initially of infection if it's communicable. Endemic is this idea that certain diseases will be expected over periods of time and in specific locations, kind of like the flu. Every year we expect X number of flu cases to happen at X time of the year. Sporadic disease is different though. Sporadic does not follow the normal patterns, so it's kind of the opposite of endemic. Either we see huge increases that we wouldn't normally see, or we see them happening in places we would not usually expect. So here's some examples. Notice here, this is Lyme disease. Lyme disease is typically found in the northeast and north central United States, but we do have random sporadic cases all over the rest of the country. And so the idea is, again, this is endemic or normal. This is a deviation from normal, so we call it sporadic. The same illustration we can see over here, but this is with the incidences of, looks like flu. So you can see every year, flu declines in the summer, spikes in the winter. Declines in the summer, spikes in the winter. But notice again, any deviation above the norm, it can be considered sporadic, or in this case, even epidemic. Epidemiology isn't just about a local or even a national level. It's even a worldwide issue. In fact, there's always going to be new infectious diseases out there that we're going to have to try to learn to deal with and ultimately even track. So HIV is a kind of good example Again, first developed in the 1980s in a very specific location, but within relatively quick amounts of time, it was starting to spread across the country and around the world. So again, this was an illustration of how we can have a new infection become very prominent, and now an epidemic that is a pandemic. There are other things we're concerned with, though, like re-emerging diseases. We're concerned with these because we at one time thought that we had controlled the infectious agent, but now we're starting to see that we're not controlling it, and that is concerning because, again, epidemiology is trying to work to decrease rates of infection across populations. There are two different things that we should talk about in terms of terrorism risk. There's bioterrorism and agroterrorism. Bioterrorism would be the purposeful use of a microorganism to make somebody sick. But agroterrorism is a little different. With agroterrorism, we're not going to try to make individuals sick, but rather the food supply. So it would be the destruction of crops and agricultural products. This would lead to, again, mass starvation and still the terror that terrorists want. And so the idea here is we're pretty aware of bioterrorism, but agroterrorism is another venue that we need to protect ourselves in.